Have you ever wondered why the cells of the human body are not completely identical if they all started out as the same cells? Animals develop by the process of mitosis, which is the process where a cell divides to yield two identical cells. As a zygote develops into an embryo, it consists of a little more than just a cluster of stem cells. A blastocyst is an early-stage embryo made up of embryonic stem cells which have the potential to become any type of cell except placenta. With 220 different types of cells in the adult body, how did the cells know which type to become? In this video, Sabi, Jacqueline, Stravani, Leanne, and I will inform you all on the basics of body plan development.
It is not the strongest of the species that survives, nor the most intelligent that survives. It is the one that is most adaptable to change. This remark by Charles Darwin captures the essence of his lifelong work on the evolution of our species. Evolution is the change of inherited characteristics within biologic populations over successive generations. Such processes give rise to diversity at every level of living organizations, including species and individual organisms.
All life on Earth has descended from a common origin that existed over approximately 3.8 billion years ago. Repeated speciation and the divergence of life can be inferred from shared sets of biochemical and morphologic traits.
In examining the evolutionary history of a species, we must look at both existing species and the fossil record. As is known, Charles Darwin was the first to formulate the theory of evolution based on natural selection. The process of natural selection infers three facts about biologic populations.
Firstly, more offspring must be produced than can possibly survive. Secondly, biologic traits vary among individuals within a population. And thirdly, variations in trait between individuals are inherited.
So what does it mean for a population if it is to survive? A key feature requires that the members of the population should be strong enough that they are able to pass on their successful traits to offspring before they expire from a natural death. As a result, such members will be replaced by progeny who will be better able to survive and reproduce in that environment, as they are genetically more equipped to adapt. In addition, this implies that there will be a higher survival rate in that specific population within a species.
The process of natural selection creates and preserves traits that are best fitted for the functional roles organisms must perform in order to survive in their environment.
Natural selection is the root cause of adaptation, but Hox genes are the basis for differentiation between different species body types. Differentiation between various morphologic constitutions is governed by the Hox genes. All animals begin with the same complement of genes that direct the development of their overall phenotype.
However, Hox genes control the segmental identity of organisms, determining the axes of formation rather than actual being responsible for the genetic makeup that determines the phenotype. For example, they set up the head-to-tail sequence and other axes of the body. You can think of them as directing instructions as an embryo develops. Put the head here, legs go over there.
In a sense, Hox genes sequence the switching of other genes. The critical function of these Hox genes would explain why a similar set of genes that is found in today's animals must have existed in the earliest common ancestor of such organisms. It comes to show that Hox genes also demonstrate how evolution has evolved in the process of vertebrate axal morphology. This is further evidence that in the mapping of evolution of organisms, differentiation in body plans for most organisms is a product of evolutionary change.
First of all cells must undergo cell differentiation, which is a process where cells become specialized in structure and function. The organism must also undergo morphogenesis, which is a process that the organism gets its form. The human genome is completely identical in all the cells, except it is just expressed unequally.
Here is the Drosophila. There are two types of genes that determine its body formation. The first set is the egg polarity genes, also known as the maternal effect genes. They determine the body axes. For example, the anterior to the posterior, which is the head to the tail, and the dorsal to ventral, which is the back to belly, and the left to right. The second set of regulatory genes are called homeobox genes, also known as Hox genes, and mutations in these genes can cause the legs to grow out of the head in place of the antennas.
Homeobox genes are DNA sequences which are found inside of genes that are involved in the regulation of patterns of anatomical development such as morphogenesis. In 1983, homeobox genes were discovered independently by Ernst Hafen, Michael Levine, and William McGinnis, who were all working in the lab of Walter Jacob Gehring in Switzerland. Homeobox genes were also discovered by Matthew P. Scott and Amy Weiner with the help of Thomas Kaufman at Indiana University in Bloomington.
On average, one homeobox is about 180 base pairs long. The homeodomain is a protein domain that can bind to DNA when it is expressed. Each homeobox chain has a corresponding residue chain. This picture shows the consensus 60 residue chain that corresponds to the homeodomain. The dashes in this chain are the introns.
Homeobox genes are a part of a class of regulatory genes that are coded for nuclear proteins. Therefore, homeobox genes are termed as homeoproteins that act as transcription factors. The homeodomain, or the HD, is the protein domain that is encoded by the homeobox. It is able to recognize and bind to specific DNA sequences. Homeoproteins are also supposed to be able to regulate the expression of targeted genes. They should also be able to direct the formation of many body structures during early embryonic development. Homeobox genes are able to switch on cascades of other genes because they encode as transcription factors. DNA is bound by homeodomain in a sequence-specific manner. However, one homeodomain protein is not enough to recognize its desired target genes. Homeodomain proteins act in the promoter region of their target genes as complexes with other transcription factors. These complexes have a much higher target specificity than a single homeodomain protein. Homeodomains are encoded both by genes of the Hox gene clusters and by other genes throughout the genome.
So the distinct group of homeobox genes, called Hox genes, found along chromosomes, are what determine the differentiation of cells and tissues, and the process of pattern formation in developing organisms. They tell stem cells what types of cells to eventually become, and when and where certain body parts should develop. In certain organisms, like flies or humans, they are located in clusters of DNA. What exactly are Hox genes? Well, Hox genes are a group of related genes that control the body plan of an embryo. The products of Hox genes are Hox proteins, which are transcription factors. They can then bind to specific nucleotides on DNA called enhancers that can either activate or repress genes that in turn conduct cellular differentiation and pattern formation. The Hox genes are able to bind to DNA with homeodomains, which are 60 amino acid long chains found on the Hox proteins. Hox proteins determine the type of segment structure formed, such as legs and wings in fruit flies, but don't actually form the segments themselves. Hox genes in mammals have 2 exons and 1 intron. During development, the genes are read in the 3' to 5' order, and their organization along the chromosome corresponds to their sequence of expression along the anterior-posterior axis of the body. You can see, Hox genes are very important. But how were they first discovered? Well, Hox genes were first discovered in fruit flies, or Drosophila melanogaster, after 2 interesting mutations were observed. In the antennapedia mutation, a leg was found where antenna should have grown, and in a bithorax mutation, the halter, which is a small knob-shaped part of the hind wings, was transformed into part of the actual wings so that the fly looked like it had 2 sets of wings.
So as you can see, this one is the wild type fly, so it has normal antenna. Well, this one is the antennapedia mutation, so that the legs ended up where the antenna should be. And in the bithorax mutation, this is the normal wild type fly, while this is the mutation, so it looks like it has 2 sets of wings.
Observing these mutations, Edward B. Lewis was able to establish that certain genes, later called homeotic genes, directly affected the expression of where certain body parts would grow. These mutations were called homeotic transformations, from the Greek word homeosis, meaning a transformation of a body part into another. Drosophila geneticists came up with the term homeotic selector gene to show that a master regulatory gene could control the development of each segment of the fly along the anterior-posterior axis of its body. About 30 years later, two scientists named Christian Nusslein-Volhard and Eric Weishass were able to identify about 1,200 genes in the fruit fly necessary for pattern formation, and eventually helped create a better understanding of the Drosophila body plan development. All three scientists' contribution earned them a Nobel Prize in 1995.
Maternal effect genes. The axis development of Drosophila is determined by genes in the egg. These genes are termed maternal effect genes. A maternal effect gene is fundamentally a gene that appears mutant in the offspring if it appears mutant in the mother, regardless of the offspring's genotype. Maternal effect genes are also coined the term egg polarity genes because they also control orientation and development of the egg itself. Because offspring develop from eggs, if a mutated gene from the mother is present, it would lead to a mutated gene product, inevitably also leading to mutation in the offspring.
An example of how maternal effect genes can affect the body axis of an offspring is seen in a bicoid mutant. Bicoid means two-tailed, and an embryo that is bicoid has a tail at both ends and no head. As a result of this characteristic, scientists hypothesize that the product of this mother's bicoid gene must be involved in determining the anterior end of the embryo. Therefore, scientists Nusslein-Volhard and her colleagues conducted experiments at the European Molecular Biology Laboratory in Heidelberg, Germany, to test this morphogen gradient hypothesis, which formally states that, quote, gradients of substances called morphogens establish an embryo's axes and other features of this form, end quote. This hypothesis is also stated in your book. Researchers discovered that the bicoid mRNA was indeed located near the anterior ends of the embryo, as predicted by the hypothesis. Additionally, they also found out that the mRNA is produced from the nurse cells, then transferred to the egg via cytoplasmic bridges, and then secured in the anterior end of the embryo. The bicoid protein then moves from the anterior end to the posterior end, confirming the fact that the bicoid was responsible for the development of the Drosophila's anterior end.
There are three benefits that resulted from this bicoid research. First off, it identified one of the earliest proteins in body development, which allowed us to gain a deeper understanding of how different regions undergo different pathways during body development. Second of all, it allows us to fully comprehend the importance of the mother during early embryonic development stages. And lastly, the morphogen gradient hypothesis proved to be essential in embryonic development in other species besides the Drosophila melanogaster. On the other hand, the paternal effect gene is only present in the offspring and the father carries this mutant allele. Sperm carrying this allele are still capable of fertilizing the egg. However, the fertilized egg would be mutated due to the mutant allele in the sperm.
To conclude this informative video, Hox genes are extremely important in the development of organismal bodies. Keep in mind that the role of Hox genes allows us to explain why there exists a similar set of genes amongst organisms. As you know, this is a result of evolution and cell differentiation. You can think of a Hox gene as a conductor of a symphony orchestra. The Hox gene can use its almighty baton to direct more noise from the flutes or less sound from the violins. Altogether, the Hox gene conductor controls the gene expression of music, creating an overall euphonious melody that is pleasing to the ear, similar to how a Hox gene influences gene expression, creating the you that you are today.